Hello and welcome to today's ECHA Science Seminar. Today we are going to discuss the assessment of bioconcentration of surface active substances in fish. And for that we invited two very senior experts in the field. That's Professor Dr. McLachlan and Dr. Stephen Droge. We record this seminar and we may publish it later on. It's live, and with your help, it's going to be interactive. So please use the chat anytime during the presentations and use the microphone during the Q&A session. ECHA does not endorse any of the tools or opinions presented here. We are very much looking forward to your feedback, though, whether it's during the meeting or later on, if you drop your feedback on YouTube. ECHA Science Seminars, they represent a series of online seminars with scientific content that's relevant to ECHA's regulatory activities, both on the human health side and on the environmental health side. And our goal is to discuss science, its regulatory relevance, and how we as ECHA can take up your findings within our mandate. So why are we talking about bioconcentration? Bioconcentration describes the process where a substance is basically taken up by an organism or partitions towards biota, and it bioconcentrates because you could say the uptake outweighs the elimination or the metabolism of the substance, and as a result, the substance may bioaccumulate. That means its concentration may increase in the organism or in the biota. And in organisms, it may result in concentrations that trigger adverse effects. That's why it's a problem. And understanding how chemicals bioconcentrate or accumulate in living tissue is essential for hazard assessment and risk assessment. And we use a combination of persistence, bioaccumulation, bioconcentration, and toxicity, as well as the combination of very persistent and very bioaccumulative to classify substances. So bioconcentration on accumulation respectively, in combination with persistency and toxicology, represents a regulatory cornerstone for industrial chemicals under REACH, for biocides, for water protection regulation, and the respective classification and labeling. So there are many groups of substances that are challenging, and one of them are surface active substances. And today we will learn how they look like and why they are difficult to work with. And we will very much get to know the state of science on the bioconcentration of these substances. And for that, we have invited Professor Dr. McLachlan and Professor McLachlan heads the research unit Chemicals in the Environment. He's an environmental chemist, and his research is mainly on how chemicals move through air, soil, water, and food chains, and ultimately accumulate in organisms, humans, and wildlife. Stephen Droge, an environmental chemist who has been very much focusing on investigating surface active substances, their environmental fate and ecotoxicology, both for surface active substances and ionizable substances. And Stephen works at the Wageningen University. Stephen has been applying both lab studies as well as modeling in order to enlighten the behavior of those substances. And now I would like to hand over the floor to our two lecturers.
Thank you very much, Jan. I would like to say a few words to start this off. I would like to start by thanking ECHA for approaching me to hold this webinar. I think that this is a fantastic initiative. And our research is often motivated by needs to improve chemicals management. And so we are absolutely delighted when we get the opportunity to address an audience such as this. And I was also delighted that I could recruit Stephen to join me today. And in contrast to myself, studying surfactants has been at the core of his research through most of his career. And he's probably the world's foremost expert on the bioaccumulation of surface active substances. And he's also excellent at integrating stakeholders into his research. And as well, the project we will talk about today was his brainchild. So Stephen will start off today's presentation, and I will chime in sometime during the second half. So Stephen, over to you.
Thanks, Michael. And also for me, it's a pleasure to present this work. A nice opportunity to showcase what we've been doing and also to update this very esteemed audience, and also the opportunity of broadcasting this presentation live and later on to explain what we are doing on the field of surfactants. The project that we will mostly discuss is funded by the CEFIC LRI initiative. The name was ECO37. We run this program for 4 years in 2016 to 2020. And at that moment, I was still working in the University of Amsterdam, and Michael was working in the Stockholm University. And the aim of the poll was to develop in vitro parameters to assess the bioconcentration of surface active substances. And for me, that was a nice challenge and continuation of what I had been working on. But I asked Michael to join because we needed high quality bioconcentration studies to verify the modeling approaches that we were looking for. That's also how we got connected. And part of the modeling was also heavily supported by the work of John Arnott and James Armitage in Canada, who joined us in the research team. So just a brief overview of the contents. I will try to start very basic on what are surface active agents, then address some key challenges in the assessment, the risk assessment for surfactants, and then zoom in on what are the key properties that we need to assess bioconcentrations for these chemicals. Mostly we will follow the project results, starting with how do these chemicals bind to cells and what kind of descriptor do we want to measure to start modeling how these chemicals bind to tissue. These chemicals may bind to tissue, but we know from previous studies that most surfactants are rapidly biotransformed by many processes in tissue, in biota, for example, liver enzymes. And because of these rapid elimination rates from biotransformation processes, this heavily influences the bioconcentration factor. There are in vitro assays that we studied to get these biotransformation rates as part of our modeling approach. Then we will pick up with Michael on the in vivo work with fish studies, where we measured bioconcentrations of several surfactants. And in the end, we will try to highlight some of the key processes as part of our modeling approach. And we end off with some conclusions and recommendations, and of course the Q&A in the end. Very broadly, nobody calls these chemicals surface active agents. They are typically abbreviated as surfactants. And surfactants are typically organic chemicals with a hydrophobic alkyl chain and a hydrophilic unit at one part of the molecule. And because of these hydrophobic and hydrophilic properties, Most surfactants spontaneously form micelles, as you can see in this globular agglomeration of different molecules in different shapes, depending also on the structure. And because of these micelles, we use these kind of chemicals also to remove dirt in our laundry scheme as detergents, for example. They do not only form micelles in the solution, but they also form accumulated layers at the air and water surface, where they are well known to also lower the surface tension of water. And so they have specific properties as this group of surfactants chemicals, where they have properties that are used in many kinds of household products, in cleaning, but also in many industrial processes. So they also decrease friction. They are, for example, used in oil drilling in large volumes. They may also optimize the spray droplets because of the surface tension decrease, and therefore they are used in lacquers. They emulsify because of these micelle forms, so they are also heavily used in paints or cosmetics or other kind of products as an important ingredient. Some examples of well-known chemicals. One is sodium lauryl sulfate. Lauryl just indicates that the chain is C12. This has a sulfonic group, but you can also have a sulfate group at the end. This is the typical ingredient of soap. The second one from the top, you can see that there is a nitrogen with a plus sign to indicate that this is a positively charged group. And as a salt, it is often accommodated with chloride, but it can also exist with bromide. One of the issues here that depending on the salt composition, you already have different CAS numbers. So looking up these chemicals may already be a challenge when you have different salts being present. Benzalkonium chloride is an important chemical as part of for example, the COVID hand soaps that are now heavily used since several years. The third compound, you can see it has a repetitive unit of 1 oxygen and then 2 carbons. This is an ethylene group. And we call these kind of structures alcohol ethoxylates. These are the typical non-ionic components of laundry detergents. In the last, the bottom molecule is an amine oxide. You can see here that this molecule has both a positively charged and a negatively charged group. These we call Twitter ionic surfactants. And also these have specific applications in specific products. Other Twitter ionics that are widely used are betaine structures, for example, in many conditioners or shampoos. So basically they all share this long alkyl chain, and often they have an ionic group to make them partly hydrophilic. Surfactants, because they are widely used as cleaning ingredients and industrial applications, they also have a potential for high emissions. In the 1950s, when washing machines started to become more common in households, the emissions were so high that rivers started to foam. And it was quickly realized that this was an emergent issue that needed to be dealt with, because the foam could also ignite on fire, and we would have real large contamination issues. The reason that it was present in the river is that the typical surfactants used in those days were non-biodegradable components, where the alkyl chain was heavily branched. You can see it in the subcaption. These were tetrapropylene sulfonates. Because the bacteria typically cannot handle heavily branched structures, the detergent producers switched to carbon sources that often contained linear alkyl chains. You can think of palm oil. And the typical readily biodegradable components are then for anionic surfactants, LAS, the linear alkyl benzene sulfonate, and the non-ionic alcohol ethoxylates that I just showed an example. And importantly, ready biodegradability needs to be confirmed for all detergent products according to the EU detergent regulation. As part of our project, we had a look into the nicely publicly available EU REACH database, and it allowed us to screen for surfactant kind of products. And one marker we entered to look for surfactants was that they lower the surface tension. So we looked for compounds with an aqueous surface tension of less than 65 millinewton per meter as one threshold. And we also only selected those chemicals that had these carbon chains of 8 or more carbon atoms long. And the result was that we identified at least 250 different surfactants with a charged group. These are Twitter ionics, about 20, anionic surfactants, about 90, and cationic surfactants, also about 90. In these dossiers, we find a lot of information. And these were summarized for cationic surfactants in the current graph that is a bit busy, but I can try to explain it. On the left y-axis, you see the tonnage band that is reported for each chemical. And each chemical is listed on the horizontal axis as a CAS number with a bar, a bin to indicate the tonnage. And the bin, the bar is plotted on the right y-axis. And the right y-axis indicates how many carbon atoms are part of the molecule. You can see this ranges from 8 up to 40, or maybe 38 or 36. For example, for surfactants with 2 long carbon chains. Another indication are the green check marks. So all the compounds that are indicated in the dossier as readily biodegradable, you can see the green check mark. These are sorted on the left part of the graph. And we go from the left to right with the lower tonnage bands, this order. And you can see that we have variant chain lengths for all these molecules. On the right part of the graph, you see that also considerable part of this domain of cationic surfactants are not readily biodegradable compounds. They are inherently biodegradable, so between 10 to 55% removal of the parent compound in 4 weeks, or even less than 10% removal of these compounds for some of these components. We hope that these inherently and no observed degradable compounds are not part of the detergent class, of course, but they maybe have specific industrial applications or other issues. The key challenges for surfactants are thus not really in how much they are produced, where they are used, or if they are degradable or not. That is well documented. The issue is often in how to predict the fate of these chemicals. For neutral chemicals, octanol-water, the KOW, this partition coefficient is the key descriptor to predict how chemicals move between water and hydrophobic phases like soil, and also like fat in tissue. And KOW is the key descriptor to predict the bioconcentration of neutral chemicals if no in vivo fish data is available. However, octanol does not really account for favorable ionic interactions that may occur between the surfactant head group and specific tissue components. Of course, fat is a hydrophobic neutral part of tissue, but cell membranes also have hydrophobic cores as the bilayer of phospholipids, but they also have Twitter ionic head groups. And these Twitter ionic head groups allow for favorable ionic interactions between the surfactant molecules and cell membranes. And of course, proteins are full of charged side groups and pockets that may also bind ionic chemicals. Our viewpoint that we started from in this project is that oxanol should not be used to predict the tissue binding of ionic surfactants. Mind you, this was in 2015 when we wrote the proposal for this project. In the meantime, chemical industry who are producing surfactants are also working with the task force in the consortium of ERASM to deal with this issue. And they looked from their point of view in how KOW can be measured in different methods, how it can be predicted with different algorithms. And it was also their main conclusion that they needed newer alternative experimental partitioning methods that have more biological relevance, and also that are methodologically defensible to describe the partitioning of surfactants in the environment. So the key challenges to model bioconcentration start with understanding the hydrophobicity for these charged molecules. If we cannot use KOW because it's highly uncertain and unable to describe favorable ionic interactions, can we use cell membrane binding as the key descriptor? But there are more issues to deal with for these ionized chemicals. For example, do ionic molecules pass the gills of fish at all? Or do they simply stick to the outside of the fish? When they are in the fish and we want to go for a model approach, how do we capture the biotransformation rate without using too many in vivo studies that are ethically non-defensible in many cases? It's not only that these chemicals are ionic, but some surfactants may also be ionizable depending on the pH. And pH may be a critical factor to determine how much of the chemical is in the neutral form and how much is in the ionic form. And that can also strongly influence how these chemicals accumulate in tissue. To capture the biotransformation rate, we focused in our project on using the in vitro liver S9 assay that was at that time just ready as an OECD guideline. And we got a lot of help from the US EPA in starting up the method and getting the right liver S9 material as a kickstart of the project. And as I said, another challenge is that if we want to model bioconcentration factors for different surfactants, we need also high quality fish in vivo BCF measurements. As I said, there are quite some bioconcentration studies available for the typical detergent structures like alkali ethoxylates and LAS. And we know that the bioconcentration factor is mostly low for these compounds because they are rapidly biotransformed. However, for example, for cationic surfactants, there was hardly any bioconcentration data for fish available to even compare our model output to any measured data. Just to briefly discuss the difference between storage lipids and cell membrane lipids. Cell membrane lipids are composed of mainly phospholipids that have these hydrophobic tails. And you can see in the picture, it forms a bilayer with the internal part as a hydrophobic domain. But on the outside, it has these Twitter ionic ammonium groups and the phosphate groups to bear both types of charges. And surfactants like to position themselves accordingly alongside the charge, the counter charge in the head group, and with the tail then protruding into the hydrophobic core. These charge groups don't like to be included in the neutral fatty part of storage lipids. We did some experiments with fish oil as a representative phase of storage lipid. You can see the graph here. On the left side, the results of a partition coefficient between fish oil and water for a non-ionic surfactant, indicated with a green symbol here. And this compares very well to the partitioning of the same chemical in a phospholipid cell membrane structure. But for the anionic surfactant that we tested here, the compound had no affinity for fish oil. It was preferred even to be in water, but it had an orders of magnitude higher affinity for the phospholipid that we studied as an artificial cell membrane. At the moment, we still ignore that these surfactants may also bind to proteins, but if you remember many of the issues with perfluorinated compounds, PFOS is also sort of a surfactant, and we know that these compounds also strongly bind to proteins. The project then focused on trying to achieve in vitro measurements and in silico calculations on the bioconcentration potential by measuring the sorption to artificial cell membranes in a standardized assay. This assay is commercially available in a sorbent dilution range, where it's packaged in this 96 well plate with a different row, and each vial contains an increasing amount of lipid material, and the lipid material is non-covalently surrounded on silica particles, and because of this heavy particle, you can easily centrifuge the system and measure what is the concentration in the supernatant, and then use a mass balance approach to calculate the sorbed concentration. As information that we get from in vitro work on biotransformation rates, we have the option of using trout liver material. There are two OECD guidelines, one using fish liver cells, and the other one is using liver S9 material. So this is a centrifuged fraction from cell material from trout. We received this from the US EPA as a frozen batch. When we activate it, we only need to add some cofactors to get all the potential enzymatic processes working. And over time, we measure how much the chemicals are degraded at different time points. I will show examples of both methods following slides. So the way we measure a distribution ratio between the cell membrane and the water concentration is this sorbent dilution series with these increasing amount of sorbent material in this series. And by spiking one constant amount to these vials, each vial will show a decrease in concentration in the water, accompanied by an increasing concentration in the sorbent material. And when we plot the results of each of these well, you can see 6 different points with red. This is an indication for C12 sulfonate. We plot it against the sorbed concentration, then the fitted line, we get as a result the partition coefficient. So it's simply the ratio between the concentration in the lipid divided by the concentration in the water. You can see here 3 examples of the same type of surfactant with a sulfonic acid, but with increasing chain length. And the sorption coefficient in a log value, you can see here, follows a typical increment of half a log unit with each additional carbon in the tail. Doing this allowed us to rapidly accumulate a wide range of surfactants in series of homologues with different chain lengths and the same head group, for example. By doing this, we created a data matrix that also allowed us to develop quantitative structure activity relationships. So we can use this data set specifically for surfactants to untangle how the molecular composition contributes to this partition coefficient. So even if we cannot measure absorption coefficient, once we have data on analog structures, we can easily extrapolate this according to simple structural features. So just to reiterate again how strong surfactants bind to phospholipid as part of an artificial cell membrane, we looked at this example from the REACH dossier of the typical soap ingredient, sodium lauryl sulfate. The REACH recommended partition coefficient that was available in, I think, 2018 in the dossier, recommended a partition coefficient between octanol and water of -2 based on the solubility ratio. So they used the maximum solubility in octanol and the maximum solubility in water and derived the KOW from that. The same dossier also reported other methods to calculate or to measure the KOW, and you can see that this already shows a high margin between these reported values. The sorption affinity for a phospholipid membrane is log 4.6. So compared to the recommended KOW, this is more than 6.5 log units higher in its affinity compared to the recommended KOW value. And this is the key underestimation that is often used when applying octanol-water partition coefficients to predict any partitioning ratio for ionic surfactants. Many membrane water partition coefficients have already been published, also ahead of our project. There was already a partition coefficient for typical soap ingredients. In 1999, 8 non-ionic surfactants were measured. In a separate work, ionic liquid cations that resemble surfactant structures were already measured in 2017, and a whole batch of cationic surfactants in 2017. And in the ECHA 37 project, we measured partition ratios between these phospholipid materials for another batch of 37 surfactants. So in total, we already have measured partition coefficients for cell membranes for 77 different surfactants, covering 26 different types of surfactants. This means that we can already predict all kinds of trends based on, for example, the chain length, and we can confirm that this is highly consistent between all the different types of surfactants that we have. We can even quantify the effect of branching of the chain or different head groups, and even the effect of fluorination is part of this QSAR approach that we can already confirm and establish. If a surfactant is not possible to measure with this assay, we can advocate that shorter homolog structures are experimentally feasible. And from that, we can easily extrapolate to longer chain chemicals that, for example, have issues of strong adsorption to the vessels or test apparatus. The first simple step that we propose is a screening tier by a concentration factor, where we do not even take potential biotransformation into account. This we call the baseline bioconcentration factor, and it is simply a multiplication of the partition coefficient for cell membranes multiplied by the fraction of phospholipid in tissue. This is the same as is typically done for the partition coefficient between water and lipid, and the lipid fraction of tissue. And instead of the partitioning of lipid, people then often use KOW. So here we simply switch to using the membrane partition coefficient and only the phospholipid fraction in tissue. For ionic surfactants, this can be simplified based on an estimated fraction of, for example, 1% phospholipid in total in fish tissue. And accordingly, we can easily estimate this baseline bioconcentration factor by using the partition coefficient in the log value and extracting 2 log units from that. 2 log units is 1%. Non-ionic surfactants, as we showed in the example between fish oil and phospholipid, also bind to storage lipids. So there we could, for example, take 5% as a total lipid fraction in fish, and then we can simply use the membrane partition coefficient to lipid, membrane water partition coefficient, and extract it with a factor 20, so this 1.3 log units. To check if this makes sense, we looked for a surfactant where biotransformation was not playing a role. And here we switched to PFOS, so a perfluorinated compound, for which we have enough evidence that these compounds do not biotransform in fish, and they are only taken up by the gills. So branchial clearance is the dominant pathway. So if we apply this baseline screening very simple model, we have a measured membrane water partition coefficient of log 4.88. So in a normal value, that is 75,000 liter per kilogram. So in this example, I used 25% of the 5% lipid as the log phospholipid fraction, not 1% lipid. Accordingly, you get to a bioconcentration factor of 950 liter per kilogram. And compared to measured bioconcentration factors in different fish, this is really spot on. We are directly predicting the bioconcentration factor of fish of PFOS based only on the membrane water partition coefficient. Of course, this is only one example for a surfactant that is fluorinated. So of course, we would like to further validate this approach in higher tier modeling with multiple surfactants. But the key is now that we can distinguish for surfactants where we are pretty sure based on this screening value that they are very unlikely to surpass the biotransformation, bioaccumulation threshold of, for example, for REACH, 2000 liter per kilogram. So you can see now on the y-axis, we plot different chemicals with their membrane lipid partition coefficient. And you can see here a whole series of different surfactants, all with a 12 carbon chain length. The green ones are non-ionic surfactants. You can see two examples with different numbers of ethoxylate units. These two green examples have absorption affinity to membranes that would not surpass the B threshold in our simplistic estimate. But we now know if the chain length is longer, how much extra carbon atoms to the chain would likely surpass the B threshold. And the same we can do for all the ionic surfactant groups that we have covered here with the C12 compound. And we can simply add up half a log unit for each carbon atom to investigate which chain length of which surfactant type would be surpassing the B threshold based only on this screening application. And you can see now in the blue box the calculated chain length for all these different surfactant types that would surpass the threshold of B, and that would indeed require a higher tier assessment of the bioconcentration potential of those surfactants. So based on only this screening bioconcentration factor using a partition coefficient, our project ECHA37 made the following decision scheme. For neutral chemicals, as long as we have high confidence in the octanol-water partition coefficient, this may also be used as a sufficient descriptor to predict how these chemicals bind to lipid. We can gain more confidence, for example, by checking alignment for different QSARs, checking different molecular fragments that are present in the molecule if we sufficiently understand that, or even overlap with high quality KOW measurements where the slow stirring method is the preferred. If this screening BCF for a compound based on KOW for neutral surfactants is below a certain trigger, we would not see a need to go to higher tier testing. If it is above the trigger, we need this bottom right corner box that will allow us to go to the higher tier assessment. For all ionic surfactants, we recommend not to use octanol-water, not to use simple QSARs to predict KOW, and also not to simply rely on a consistent offset factor between the neutral and the charged form. Instead, we advocate that these membrane water partition coefficients are a first key descriptor that are experimentally feasible and also mechanistically sensible to do. To continue with the higher tier assessment, we have several publications coming out of our project. that showed that we can do in vitro assessment on biotransformation rates for cationic surfactants using the rainbow trout liver S9 material, and the same for anionic surfactants. This has been sufficiently described. There are in silico tools that can support us and also modeling framework to make sense of how we link in vitro assessments towards predictions on full fish bioconcentration factors. And for the in vivo work, I already list here some of the publications that were the main output that Michael will present further on in the presentation. Just to briefly present some of the work we did on the in vitro assays on the liver trout S9. For each surfactant that we tested, we used a pure homologue with a known chain length. We compared always inactivated trout liver enzyme fractions that we call inactive S9 to verify that the chemical remains in solution over time. You can see here on the x-axis, the time scale can be extended up to about 2 hours in this assay before the enzymes lose their activity. But for the active S9, if the measurements go well and the chemicals undergo enzymatic degradation, we can see on a log scale a nice linear decline in the concentrations due to biotransformation. And from this, we can derive the biotransformation rate that we can then apply in modeling approaches. Of course, we tried to pair as many of these clearance rates to the available partition coefficients that we already measured for surfactants. And we tried to, of course, pair this for as many surfactants for which in vivo bioconcentration factors were already available, or that we were planning to test in our in vivo studies. What we learned from these biotransformation assays is that there are some trends, but the trends never point in one direction. You can see on the left graph a set of different non-ionic surfactants with increasing chain lengths from left to right and with different number of ethoxylate units. So there is some kind of optimum visible where compounds show a high biotransformation rate, and typically at longer chain structures, we see a decline in the biotransformation rate. For cationic surfactants and anionic surfactants, we also see there is some kind of optimum, but then again, there is also large variability between the different types of hydrophilic head groups in how they are amenable to the enzymatic degradation rates. The other issue that we encountered is that there are limitations in the detection rate of significant biotransformation in our assay. We can only run it with active material for 2 hours. And ideally, we would also like to monitor if low biotransformation is still visible over a longer time period of, for example, a full day or 2 days. But this is not yet possible with the S9 material when we were testing it. But that doesn't mean if we don't see a significant degradation rate that the compound is not biotransformed at all. Here is a first example of how we put the in vitro parameters together in a modeling approach to predict in vivo bioconcentration factors. And as a case study, I highlight here the non-ionic surfactants, typically are easier to model, where we understand more on the partitioning and have less issues with, for example, gill permeation. And we already had high quality bioconcentration factors and partition coefficients for the membranes. What we added now was the biotransformation rates that we measured with liver trout S9. You can see the raw data here in this graph. But when we tabulate these values with the measured bioconcentration factors by Johannes Tolles in 2000, and the partition coefficients measured in the Swiss group, we pair it now with the clearance rates on our S9 material. And we have for some of these structures, a measurement. And the question was now, can we extrapolate the compounds that we did not yet measure with some values of an in-between clearance rate? When we do so, and we apply these rates in a model that allows us to make the in vitro to in vivo extrapolation. And one model we used is the Bionic model, which is part of the EASY Suite assessment platform by the Arnaud group. We can make sense of how much the clearance rate is contributing to the lowering of the bioaccumulation factor. On the right column, you can see what our partition coefficient would predict for the baseline concentration factor. And you can see the difference between the last two columns is then the impact of biotransformation. In a plot that looks like this, on the x-axis, we now have the predicted bioconcentration factors with the model. On the y-axis, we plot the experimental bioconcentration factors. All the open symbols are the screening tier bioconcentration factors, and you can see that some would be predicted to be in the range of bioaccumulate, bioconcentrating, or very bioconcentrating. But based on the in vitro clearance rate, the model now shows how much we lower the bioconcentration due to biotransformation. And you can see now with the closed symbols, the modeling output. It seems to be in quite a narrow range on the predicting range, but it's still scattering within a factor of plus or minus 3 for most of these compounds around the in vivo data that we have. So to indicate the power of this modeling approach based on a partition coefficient and an in vitro clearance rate. So as an in-between summary, we have a lot of measurements for this membrane water partition rate already for surfactants. We have for many surfactants already in vitro clearance rates based on single compound tests, as well as confirmation of some of mixtures that have or have no influence on this biotransformation rate. We also confirmed several formation of metabolites in these in vitro studies. And that allows us to now compare these model outputs for measured bioconcentration factors. And what we will show in the next section are the results of in vivo tests with cationic compounds and 11 anionic compounds. And this is where I switch to Michael to continue with the presentation.
Good, then I will continue now to talk about the in vivo studies that we have done. And as Steve had indicated, in vivo studies were necessary for us to be able to check if our knowledge that we derived then from the in vitro studies and the distribution studies can actually be applied in vitro. So we started out by studying cationic surfactants, largely because of the paucity of data that were available for these chemicals, and also because there were indications from the baseline work then that they could indeed be bioaccumulative. The first issue that we decided to address was to look at where these surfactants are actually stored in the fish. And there had been indications in the previous work that had been done that surface absorption was particularly important for cationic surfactants. And we started out then by doing a tissue distribution study with 12 cationic surfactants. And we selected these chemicals. All right. We selected these chemicals then to cover a range then of different chain lengths and also a range of different head groups. So we looked here at ionizable alkyl amines, and we had both primary, secondary, and tertiary, as you can see here. And we also included then quaternary ammonium compounds as well, so permanently charged cationic surfactants. And here we exposed then large rainbow trout, about 100 grams for a week, and then we dissected them and we explored the tissue distribution. And what you see here is the different tissues that we studied and the fraction of the residue in the fish that was present in each of these tissues. We looked at both surface tissue, mucus, and also at skin and gills, which are exposed both via the surface and internally, as well as internal tissues here, muscle and liver. And here you can see a typical result then for the ionizable alkyl amines, so those compounds which do dissociate. Here we find then a significant fraction of the residue associated with the surface tissue. Here, mucus being a pure surface tissue, but also a significant fraction being associated with the interior tissues. Looking at the quaternary ammonium compounds here, on the other hand, we see that the residues are almost entirely associated with the surface tissues, with mucus and with gills. And the skin, liver, and muscle fractions that we see here are actually based on LOQ values. We were not able to actually prove that the chemical was present in those tissues. So both surface and internal accumulation is important then for the ionizable alkyl amines, and for the permanently charged substances, it looks like that they are not being taken up systemically. We then continued to do a study then for the same 12 cationic surfactants, then a BCF study. There we had a 2-week exposure period by a 4-week clearance period. We did modified OECD 305 kind of study. We did them in these large basins here with juvenile rainbow trout in the order of 10 to 20 grams. And you can see here typical results of what we found. These chemicals, they ranged in chain length and from C9 to C16. Particularly at the longer chain lengths, there were concerns from the literature that BCFs were actually not measurable because of absorption effects. We did quite a bit of work to optimize then both our exposure system to minimize then absorption sinks within the aquaria, and also with the sampling and analytical procedure to minimize the possibility of absorption loss effects then. And the results that we got were very satisfactory. We were able to maintain concentrations then throughout the experiments, throughout the exposure phase. And you can see here a typical result then here for T, so for the tertiary amine with a carbon length of 14. Here we ended up then with an in vivo BCF value of 8,400. And the comparable value then for the quaternary amine ammonium compound, the C14, was 52, so more than 2 orders of magnitude lower. And then when we return to Stephen's work and we look at the screening baseline values, we see that the in vivo value is about a factor of 4 lower than the screening value based on partitioning than for the T14. And for the quaternary ammonium compound, our permanently charged cationic structure, we have a value which is 2 orders of magnitude lower. When we then progress to the higher T tier estimated BCF, Here we see that the agreement, which was not too bad then for the T14, when we actually look at the higher tiered value, which has been corrected then for biotransformation using the in vitro S9 assay, we get a value there that is actually much, much lower than the in vivo measurement. So here we have a significant danger of false negative values. Whereas then the in vitro S9 then indicated no biotransformation then occurring then for the Q14 compound. And here we can conclude that the quaternary ammonium compounds are showing much lower than expected bioconcentration factors because they are not permeating through the gills and entering the systemic tissue. We did these studies then at 2 pHs, at pH 7.6 and pH 6.2. Here we have a summary of the results then for the pH 7.6. And here we see for 4 different chain lengths, the results then for our tertiary amine, and we can see that there is a significant increase in BCF with chain length. And we can see that some of the BCFs actually exceed the B threshold. When we then include the primary, here in blue, and the secondary amines in orange, we can see that for the same chain length, we generally have lower values for the primary than we do for the tertiary. And there are indications that this is also the case for the secondary. We see some evidence of a flattening off here that had higher chain lengths that might be due to bioavailability or absorption effects than during the experiment. This is something that requires further investigation. And when we plot in the quaternary ammonium compounds, we see these much lower values. And here we see then that there is something fundamentally different happening here. And as we indicated, then that this appears to be due to poor permeation. Here then a summary of what I just said and the paper in which it was published. This plot then summarizes then the comparison then between the in vivo values and our model predictions of what we would get, assuming then no biodegradation, so essentially the baseline screening value, and assuming biotransformation using the S9 in vitro study results. And these red boxes here, they illustrate the discrepancy between the in vivo value and our higher tier estimate. And we can see that it is quite considerable for the amine surfactants. We have an inconsistency here between the biotransformation rates that we are getting from the S9 data and the in vivo results. This is something that remains unresolved, why the S9 overestimates the in vivo biotransformation of these compounds. This is something that we need to study further, but it's also an illustration of the value of in vivo studies to actually verify what we derive then from in vitro work. Here the B and the BP thresholds again, indicating the exceedances. We continued this work then to look at anionic surfactants. Here we studied 11 different compounds using a similar kind of study. And to briefly, here you can see some of the results as well. A brief summary here of those results at the bottom. You can see that we had faster elimination of the anionics than we did of the cations. So we could do shorter experiments. We were often approach steady state conditions during the experiment. They were less stable in the water than the cationics. We had a little more trouble maintaining stable concentrations. And when we took our data and compared them with the one strong data set in the literature that actually has good BCF values for anionic surfactants, that's the work of Johannes Toos, we actually got good agreement, which was very reassuring. And here you can see, again, a comparison of the effective structure on the bioconcentration factor for the anionic surfactants. Here we have then a series of 5 different sulfonates that were among the 11 compounds. We see a very strong and pronounced chain length effect. And then we can see some indications that the sulfates then tend to have somewhat larger BCFs than the sulfonates, and the LAS then even larger again. All of our test compounds lay below the B threshold, but there is some indication that it might be possible, for instance, then for a C16 sulfate, that it might actually exceed the threshold. So some conclusions then briefly from our in vivo experiments. It is possible to do in vivo experiments for surfactants and to get high quality data. I think we could show that. Care was needed with water sampling and analysis, and also for the anionic surfactants and trying to minimize the possibility of biodegradation in the experiment. Really important, I think, is that it's possible to do multiple chemicals in one experiment, and that can greatly reduce the number of animals that are required to get this information when it actually is required. And also building on Stephen's work then, when we do these experiments for ionic surfactants, then there is no correction done for lipid content, because it is not the lipid that is the major repository of the chemical. It is the phospholipids, the membranes. So it's not the neutral lipid. Moving then back to our anionic surfactants and comparing it with model predictions, and these are model predictions that were made with the model that James Armitage and John Arnott developed within the project. We can see with the open symbols, the predictions that were made then using a screening tier assessment. And when we actually then corrected the predictions then using the biotransformation information from the in vitro studies, we can see then that our open symbols then move to the left, and they then agree much better with the experimental values, approach the one-to-one line. And this indicates for the anionic surfactants, we actually have the in vitro work and the distribution work aligning very well with the in vivo data. Returning then to the tiered screening approach and how we can actually build a decision tree for assessing the BCFs of surfactants. Here we see the screening tree that Stephen produced, and if we then progress then using the results from the in vivo work to build a similar tree for the refined BCF assessment, here we can see we have, if we start here, we select a somewhat conservative threshold of the BCF baseline of 500, and we say if the chemical exceeds the baseline value of 500, then we're a little bit uncertain as to whether it might possibly be B. So we then proceed to refined assessment. If the chemical is permanently charged, such as the quaternary ammonium surfactants, then we would likely conclude that it is not B. We have not explored, of course, a very broad space, chemical space for the quaternary ammonium surfactants. This remains to be done. If not, then if it is an anionic or a non-ionic surfactant, then we can use the in vitro data with all the lines of evidence that we can collect from that and our models then to predict if the BCF will be greater or less than 2000. And if the prediction is true, then it is likely to be B or VB, and then one can consider doing a test then to further explore that. For the amine-based surfactants, then we cannot, at this point in time, use the in vitro S9 approach. And there we can nevertheless make some estimates based on our results that if the chain length is greater than C12, then there is a possibility that it will be bioaccumulative. And if it is greater than 15, there's a possibility, good possibility that it will be very bioaccumulative, and then that further testing would be indicated. Whereas if the chain length is less than 12, then this is unlikely. And finally, for the Twitter ionic surfactants, we don't have any data at this point in time that allows us to progress beyond the baseline assessment approach. So, excuse me, moving on from there, we now have a situation where we, based on a relatively limited number of chemicals, have developed these screening approaches that we can use for exploring the BCF of these chemicals. But of course, we encounter then many different structures, and we have to try to extrapolate the results from one structure to another. To assist in this process, then, we constructed a mechanistic framework that provides some insight into what processes are controlling the bioconcentration of a surfactant, and therefore helps us in understanding, depending on the properties of the surfactants, how this BCF is likely to change. Let me try to explain this here. We start with here a mechanistic model then of transport across the gills, where we have a series of resistances starting from the gill ventilation resistance, so the actual effort that the chemical needs to overcome then the resistance in the gills and to actually get to the surface of the gill tissue, then the resistance for transport through the gill tissue, and then the resistance within the gill itself then for the actual transport of the chemical out of the gills with the blood and into the remaining tissue of the fish. To describe these processes, then, we essentially established that we could describe the uptake, elimination, and BCF of surfactants in fish with a combination of essentially two different parameters: the fraction neutral of the chemical in the water phase and the distribution coefficient between the membrane lipids and the water, shown here on a log scale with the different colors. It's also a function of the pH, which of course plays back into the neutral water. So we really have three parameters. I will show you an example here based on a gill pH of 8.1, which corresponded to the experimental data that we had with a water pH of 7.6. And here you can see then that for different values of the distribution coefficient, depending on the fraction neutral in water, we get different values here of the uptake rate constant of the chemical. What we then do is we can within this chemical space here, we can map on what the dominant resistance is, and it tells us then what the dominant process is that is controlling the uptake of these chemicals. We can then choose the chemical class that we are interested in. And here I have chosen then a typical fraction neutral range for the alkylamines that we studied in this project. And here, if we look at the alkylamines, we can see that if the alkylamines have a very low DMLW, so membrane lipid water distribution coefficient, then the dominant resistance to the uptake of this chemical will be the blood. And then, as a result, the I'll stop over that. Whereas for other higher DMLW, so more strongly sorbing chemicals, which again would be more interesting, more likely to be bioaccumulative chemicals, we can see it is the water resistance which is dominant. We can see regardless then of the value of DMLW up here, and regardless of the fraction neutral, these chemicals will all be expected then to be taken up at the same rate. So it is quite safe for this class of chemicals to actually read across then between chemicals with varying DMLW values and also varying pKa values, because they should actually all have the same value. So we've created this structure which actually facilitates the understanding and the extrapolation of BCF values, uptake rate constants, elimination rate constants, and BCF values. And a further thing which came out of this modeling work is that the pH dependence can be also predicted using these tools. And we saw with our in vivo work, I did not present that today because of time restrictions, but we saw a very clear dependence of the BCF for the cationic surfactants, the alkylamines, on pH. And here you can see how the model predicts that it will vary. So we can see between a pH of 6, for instance, and a pH of 8, we expect an order of magnitude variation then for basic chemicals with this combination then of properties. And that is quite significant, and that is something that needs to be taken into consideration, I believe, in a regulatory context as well. So we'll move on to a few conclusions here, relevant then for surfactant BCF assessment. First, we can note based on the in vivo work that the BCF can be above the regulatory thresholds. We saw that for the alkylamines. Second, Stephen was very clear, I think, and convincing in showing you that there are options to assess the potential for bioconcentration without using KOW, I should say, but instead using the membrane lipid water distribution coefficient, and that this can be a conservative BCF baseline screening tool. Biotransformation, as we saw then for the anionic surfactants, may strongly reduce the BCF. And so it is important then to include the S9 clearance information when it is available. It moves the data then towards the observed BCF. This is a very useful approach. We can try to adapt then to the S9 limitations. We have still situations where the S9 is not sensitive enough, and we have an LOD which is so high that it still may influence the BCF. Here we need to do some further work. And there's also possibilities to further optimize then this using alternative tools and species then to the S9 rainbow trout cell lines that we were using. Finally, from the in vivo side, it's possible to use far fewer animals than to get high quality data, but we suspect that we have a chain length limit of around C16. And we conclude also that it is important that we pay attention to pH, and that read across is also in many cases possible, that it's helpful in doing read across then that we get information for data rich homologs, and that we can support our read across efforts with mechanistic models. And with that, I will close a little bit behind schedule. And
thank you very much, both of you, and thank you to the audience for making this event today interactive. And the live audience, so you will receive an invitation for feedback. Please give us feedback, the lecturers and the organizers. And if you watch the video online, add a comment underneath the video. So with that, I would like to thank everyone for very insightful and very interesting presentations by our two senior lecturers. And I would like to wish you good afternoon. Thank you. Bye.
Thanks, everybody.
thank you very much, and Merry Christmas.